Thank you. First, I would like to thank the organizing committee for letting me presenting this work. And it's about, as Mr. Chairman said, the pathogenesis, host innate immune response, and aerosol transmission of the influenza D virus in cattle. So, the influenza D virus, or simply IDV, is the most recently discovered genus of the Orthomyxoviridae family. The influenza D virus has a worldwide distribution, and to this date, the proven host for this virus are mainly cattle, in addition to swine, small ruminants, and maybe camelids. We have pieces of evidence suggesting that camelids may be host for the IDV, and ongoing research is carried on now for trying to find other possible hosts for this virus, especially among wildlife. So, in most cases of IDV detection, the tropism was mainly respiratory. This respiratory tropism raised the question, many questions, mainly about the pathogenesis of IDV in cattle, its transmission, and its contribution in the bovine respiratory disease. So, in order to answer these questions, we conducted an experimental infection. In our study, we infected a group of 8 calves with a French strain of IDV with a load of 10 to the 7th TCID 50 per calf. This infection was done using a nebulizer mask, and in addition, 3 meters away from the infected group, we added a control contact calves, 3 calves, in order to evaluate the transmission of IDV by aerosol. So these calves were not infected. In addition, in a separate building, we also included a group of 5 non-infected negative control calves. Our daily monitoring of these calves included physical examination, of course, twice daily, and in order to evaluate the transmission of the virus, we did nasal swabs daily and bronchoalveolar lavage frequently, and also we gathered air samples throughout all the infection. Actually, it's every other day. And in addition, at day 8 post-inoculation, we euthanized and necropsized 3 calves. And the rest of the animals were euthanized and necropsized at day 22 post-challenge. So as a major result, we noticed that IDV induces mild to moderate respiratory clinical signs and pathology in both the upper and lower respiratory tract. These clinical signs included repeated spontaneous coughing, abdominal dyspnea with increased respiratory rates, in addition to abnormal lung sounds. But no consequence on appetite, general states, nor hyperthermia were recorded. As we can see in this graph, the clinical scores, the daily clinical signs actually, were scored on a daily basis. And this blue curve represents the daily clinical scores of the infected group, the calves of the infected group. It's a mean, actually. So we can see that the clinical signs were concentrated between day 7 and day 10 post-infection, with a peak on day 8, and with a significant difference between the infected group and the other 2 groups. Concerning the 3 calves euthanized and necropsized at day 8 post-infection, and at the macroscopical findings, we found patchy area of atelectasis with deep red textures in cranial lobes of the lungs. And microscopically, lesions corresponding to subacute rhinitis were found on the nasal mucosa. In addition to lesions corresponding also to subacute bronchointerstitial pneumonia found in the lungs of the infected calves. Furthermore, we evaluated also the replication of the virus in both the upper and lower respiratory tract. as we can see here, the blue curve corresponds to the mean shedding of the infected calves, and the shedding lasted till at least till day 15 post-infection, with a peak on day 4, having a load, maximum shedding load of 10 to the 6th RNA copies per microliters. And concerning the lower respiratory tract, the shedding lasted, well, looking at in the bronchoalveolar lavage, the shedding lasted till day 14 after the challenge, with a peak shedding of 10 to the 2nd RNA copies per microliters. Confirming the lung tropism, this slide of immunohistochemistry, we can see the IDV antigen deposition on the bronchial epithelium of one lung, thus confirming the pneumonial tropism of the virus. We further evaluated the aerosol transmission of IDV, and as I mentioned earlier, we gathered air samples, first from the contact area, then intermediate area, and in the infected area every other day during the length of the experiment. And as you can see here, at day 3, we detected the RNA, the IDV RNA genome in the aerosol infected area, and by day 5 we could detect them in all 3 areas, and by day 13 post-challenge we detect the virus only in the contact area. Furthermore, if we look at this graph corresponding also to the shedding of all the groups, where the blue curve represents the infected calves, here the green curve represents one calf, the shedding in one calf of the contact area that started shedding by day 11 post-infection. Thus, and the 2 other calves actually started shedding several days later. This findings confirm the aerosol transmission of the virus to at least one calf of the contact area. Concerning the antibody response, and using the HI assay, hemagglutination inhibition assay, we started finding antibodies by day 10 post-challenge, and also using an IDV ELISA, specific IDV ELISA detecting both IgG1 and IgG2 antibodies. We also detected the antibody IgG1 by day 10, but no IgG2. We think that it might be due to the short length of our experiment. So in the search for IDV-specific cellular memory responses in peripheral blood, we gathered systemic blood at day -1 and day 22 of every calves, and we analyzed the proliferation of IDV restimulated PBMCs. Overall, the IDV-specific proliferation was significantly stronger at day 22 compared to day -1 in at least 2 calves. This calf and this one. So every couple of columns correspond to one calf here. Being detected in the lungs, we also analyzed the local immune response in BALS, in bronchoalveolar lavage, and using a transcriptomic assay, we showed that IDV infection induced an active and significant overexpression of PRRs in the lungs. You can see here it's day 2, day 6, and day 14 of every parameter. But although this overexpression was clear, we do not observe an overexpression of genes implicated in the interferon 1 pathway. This finding raises the question about if the IDV virus can modulate the immune response, at least the interferon 1 pathway response, during the infection. On the other hand, the IDV infection was able to stimulate an overexpression of inflammatory chemokines. As we see here, we have an overexpression of for example, CCL2, CCL3, CCL4, and several others. So, as a take-home message, we can say that IDV induces moderate respiratory signs. It can replicate in both the upper and lower respiratory tract. It can be transmitted via direct and aerosol routes, and has the capacity to induce a local immune response and overexpression of chemokines and a systemic cellular response. Now, the main question is this agent a predisposing factor to bronchopneumonia in calves? Well, in the field, previous studies showed that it is more likely to detect IDV in cattle with respiratory signs than in healthy cattle. We therefore believe that IDV may play a role in the bovine respiratory disease, but not as a single agent, infecting agent, but maybe in combination with other factors. So future and ongoing studies actually aim to define those other co-infecting agents and to reveal the implicated mechanisms. Thank you for your attention. Bye-bye.